Hey everyone, this is a short-ish video following up with our adaptive immunity lecture. So I'm going to start with antibodies. Antibodies are proteins. They are proteins that are associated with B cells. You have millions of B cells, and each of those B cells produces its own unique antibodies. And those antibodies, because they are unique, some of them may just by chance match up with an antigen that corresponds to a particular pathogen. So how does that happen? Well, here is the structure of our typical antibody. It's made up of proteins. Proteins are made up of amino acids. And you can see that these end regions here are the antigen binding sites. So this is where an antigen of a pathogen would match and stick to. These ends are variable among antibodies of different B cells, because these regions of the peptide chains or the amino acid chains are highly variable. That just means they change. One side is what we call the heavy chain, one side is what we call the light chain, and both of these at the end are variable areas. But as we come in, we notice that they are no longer variable. So these segments here will be very similar across all antibodies in B cells. You'll see, if you look closely, that there are also binding sites for things like complement and even mast cells and basophil cells. These will simply help antibodies to be even more efficient and for our immune system to be able to target whatever pathogen or antigen we are going after. Also important to note that antibodies have two binding sites. When an antigen binds to both parts of the antibody, each of these areas is a determinate site, and so when it's bound to both, we say it's a complete antigen, and when the antigen is bound to the antibody, we call that an antigen-antibody complex. On page 782 in your book, you can see another picture of this where some antigens will only match up with one part with one antigen binding site on the antibody, and we would consider that an incomplete antigen, sometimes known as a hapten, but I'm really more just interested in you knowing that complete antigens mean they are fully binding to the antibody. There are different classes of antibodies, and those classes are determined by the heavy chain segments. Remember, that's the purple part here. So in each class of antibody, this purple chain would be the same within that class of antibodies. We're going to have the IgG antibodies. They actually make up the majority of antibodies that are hanging around, and they will go after viruses, bacteria, and toxins. So without them, it would be very difficult for us to fight those things off. IgE actually is going to be involved with binding to mast cells, which in turn release histamine, which you might remember from the inflammatory response, causes a number of effects. And we see an increase in IgE with allergic reactions. IgD is found on the surface of B cells where antigens can bind, and that binding will lead to sensitization of B cells. And remember, B cells have to be sensitized in order for helper T cells to activate them so they can become plasma cells and produce antibodies. So IgD plays an important role there. IgM is actually secreted immediately after an antigen is encountered, but thereafter we see its concentration start to fall off because IgG starts to rise and accelerate, and so IgM isn't needed anymore. Just as a little flashback to in lab when you learned about A and B blood types and how you can't mix them. IgM is the antibody responsible for that agglutination of incompatible blood types. IgA is found in glandular secretions and like mucus, tears, saliva, that kind of stuff. And they're really good at attacking pathogens before they ever even get into our tissues, is the idea there. So here's a quick little question for you. Immunoglobulins that are most abundant and are responsible for resistance against many viruses, bacteria, and bacterial toxins are And hopefully you remember that that is IgG. But of course, you need to know what the other ones do as well. Okay, so that brings us to the idea of primary versus secondary response in antigens. So when you are first exposed to something, to an antigen, you can see that IgM rises first. And then right after that, we start to see a rise in IgG. And as IgG peaks, IgM is already falling off. But look what happens when we have a second exposure to the same antigen. Now, both IgG and IgM are going to increase, but IgM isn't ahead of IgG anymore. Instead, IgG is going to rise much more quickly. IgM will rise a little bit and then fall off, but IgG is going to have a really big response. This is going to be coming from those memory B cells that have seen the antigen before and already have the antibody that they can release in order to target. So this is a much quicker and much more robust response. This is the reason why we are so good at fighting off infections that we've already seen before. But over here, what we're seeing is how it takes some time for us to fight off something that we haven't seen before. So how do antibodies destroy our target antigen anyway? Like, what do they actually do? So they can neutralize it. When they neutralize it, it means that we're going to see antibodies can bind to sites on the bacteria or the virus, and then they actually make the virus incapable, or the bacteria incapable, of attaching to the cell itself. So we've neutralized it from being able to infect its target cell. Precipitation and agglutination, this is where antibodies bind to the antigen and then they bring large numbers of antigens together, creating a big immune complex. And when you get this big complex, it's going to precipitate out of the fluid that it's in, maybe the blood. And then also, if you get really large antigens on the surface of a cell or virus, you can form these really large clumps of agglutination of those cells or virus particles. So precipitation and agglutination are kind of the same thing, but agglutination is when they actually stick together, and precipitation is when they're so stuck together and so big that they come out of solution. They can prevent pathogen adhesion. So literally, some of our antibodies that are dissolved in our saliva, for example, can act as a covering that make it difficult for bacteria or viruses to adhere to the body's surfaces. And if the pathogen can't stick to a surface, it can't infect it. Sometimes antibodies may promote inflammation by causing the release of chemicals from mast cells and mast cells and basophils, because remember, they have binding sites for those cells on the antibodies. And antibodies can also activate the complement system. And of course, complement is really good at helping to stimulate the innate response system. We can see an increase in opsonization as a result of antibodies. So phagocytes can bind more easily to antibodies and even also complement proteins that may be stuck there as well. And that's going to increase the effectiveness and the likelihood of phagocytosis. So that's the definition of opsonization. And then the other thing is antibodies just attract phagocytes. So any antigen that's covered with antibodies is going to attract eosinophils, neutrophils, and macrophages. Is one of those chemotaxic behaviors that we see with phagocytes. So a lot of different ways that antibodies can actually destroy whatever the target antigen is. And just a reminder, antigen is basically a substitute for whatever the pathogen is. The issue is that antigens are often just pieces of the pathogen, just usually proteins. Remember that hypersensitivities are just abnormal reactions to antigens. So they are foreign. They don't belong in your body, which is why your immune system is being like, wow, what's going on here? But they're things like pollen, which actually isn't going to do us any harm. But in some people, our immune systems aren't good at recognizing that. And so we develop an allergic reaction. If it's just localized, it may be annoying. If it's systemic, it could lead to anaphylaxis. And anaphylaxis could cause us to go into anaphylactic shock, which is is a life-threatening emergency. That's what EpiPens are for. So you can see over here in a first exposure, we've got our little allergen fragments being presented by a macrophage, which is activating a T helper cell. Also, it's binding to a B cell, which is causing it to sensitize. The T helper T cell and the sensitized B cell can now come together, and the helper cell will activate the B cell, so it starts producing antibodies against that pollen. So now if you're exposed a second time, right, it's going to cause all of those IgE antibodies that are out there are binding to our mast cells and basophils. And so when they get stimulated by that allergen binding to that antibody, it's going to cause a release of histamines and leukotrienes and probably prostaglandins, all of which are going to cause inflammation and pain and stuffiness and burning eyes and itchy nose and all that kind of stuff. Some other immune disorders are going to include autoimmune disorders like rheumatoid arthritis, type 1 diabetes, or lupus. These are all diseases where your immune system is mistaking your regular cells for pathogens or foreign cells, and so it's attacking them, and the consequence are these diseases. Obviously with type 1 diabetes, it's because we're attacking the cells in the pancreatic islets, and as a result, there's not enough insulin being produced. In rheumatoid arthritis, we see elevated inflammation and joint destruction as our immune system attacks our connective tissues at the joints, for example. We also have the issue of transplant rejection. So when you put an organ into someone, the problem is those are foreign cells, and cytotoxic T cells are going to recognize those cells as foreign and want to destroy them. So it's going to require immunosuppression to try to shut those cytotoxic T cells down. We often use cyclosporin as the medication for that. But the problem is it also, the reason why it works is because it shuts down helper T cells and cytotoxic T cells, while leaving regulatory T cells relatively unaffected. And remember, they tamp down on the immune response. But that also means that people that are on immunosuppressants are at risk of infection. So you may save the organ, but they could get an infection that they can't fight off. I mentioned in class that we have diseases like HIV, which target helper T cells, for example, and so this causes immunodeficiency. So without your helper T cells, you're not able to activate the antibody mediated cascade, which makes it really hard to fight off bacterial infections, and so these patients are more likely to die of opportunistic infections. Also, just a reminder that we talked about previously, we also see that our thymus involutes with age. So as we get older, we have fewer T cells, and with fewer T cells, there's less B cell activation, and that does also lead us to an increased vulnerability to infection. Okay, and I threw this slide on here at the end because it kind of brings it all together. So this is a great slide to see how innate and adaptive systems work together to provide you with immunity. So this is a great slide for you to kind of review and talk yourself through, right? So over here, we can see here are our antigens. We can see the innate or non-specific responses, and we can see our adaptive specific immunity. Notice there's the NK cells and macrophages of our non-specific immunity, but they are antigen presenting cells. So they'll chew up anything, but then they can showcase those antigens via MHC2 proteins to our adaptive immune system, where we can get activation through MHC1 and MHC2, and then you can walk yourself through the flow chart of what happens with that activation. And then over here, we can also see how we might go after a bacterial infection versus how we might go after a viral infection. So you can see that typically we're going to have phagocytosis of bacteria by macrophages and APCs, which will then allow them to present that antigen, which will activate our cytotoxic T cells and our helper T cells, which eventually will lead to the destruction of the target pathogen. With the viruses, the key there is that they kind of hide inside our cells because they take over our cells in order to make copies of themselves. So we are going to need those antigens displayed, which is what's happening here. So the infected tissue cells may display antigen via MHC1 proteins, which would then lead to this cascade of events, or APCs may find some of those viral antigens and will go through this cascade. So again, this is just a great slide to kind of bring it all together. All right. And that's it for the immune system. Woohoo!